Okay, let's begin. So, I'm going to start with the title, Investigating Genetic Relationships, and this follows on from the taxonomy lesson because, as I told you last time, based on new evidence, we've had to actually change some of the classification systems. We've made 3 brand new domains because of advances in science, alright? And this leads me to have to tell you all how we have come up with a classification system, how we have grouped individuals in the places we have, alright? So, I'm going to start with how we used to do it and go on to how we do it today, alright? So, in the past, and this when we go back to our mate Carl who?
Linnaeus.
Good. Observable characteristics. Alright? Carl was way ahead of his time, but he still based individuals as much as he could on, there was the evidence of the evolutionary tree, but mainly on the observable characteristics. Alright? We've talked about the downfalls on that, but what he assumed was that if they had similar characteristics, they must have had similar DNA. Alright? And the problem with that, we need to be aware of the problem with that, is that most characteristics are actually polygenic. Alright? That means they are controlled by many genes. Okay? And polygenic features are very influenced by the environment. Alright? So there is a high environmental influence. Which means the actual feature itself wasn't purely based on DNA. Right? Its structure, how the growth of it could have been affected by the environment. A lot. Because most features are polygenic. Alright? Controlled by many genes. So as science has evolved and our knowledge has evolved and we've started to be able to study DNA at its base level, we have now moved on to actually study relationships in the DNA. Okay? So we actually take genes from separate species and compare them. Okay? So in the classic example, we always try to see how closely related we are to monkeys, chimps, orangutans, and so we will take the DNA sequence for a specific gene from all the organisms we're looking at, and we'll compare them. Okay? And what I've got on the board, and in a picture here, for those that are watching, is an example. Okay? An example of a DNA comparison. How this works is, they will colour with a dye the 4 different bases in DNA, which are
Adenosine,
adenine, thymine, guanine, and cytosine. Okay, and each one of those has a colour. And for life, I can't remember which the colour is, but by comparing the strands and seeing how close the colours and order of colours are, we can look for similarities between species. Okay? So we actually physically look at the DNA, we look at the order and sequence of the bases for a given gene and compare similarities. Are we alright with that? Okay, so that is our DNA relationships. Let me put that one there. We look at a gene, we compare species, and we are using a dye to look at base sequence. Now, following on from that, and it's really just almost exactly the same as sometimes we don't analyse DNA, we analyse mRNA. Now I've been looking around for the reason why we might do this instead of this and I can't find it. Alright, me and Mr. Capsick have had a debate about why it might be, and between us we haven't really come up with the answer. But the concept could be this is single-stranded, therefore easy to manipulate, Or it could be this is, occurs in the cytoplasm and this is the nucleus, therefore it's easier to extract. Alright? But it's exactly the same. These mRNA has bases. The bases will be comparable in different species, and therefore the more similar the mRNA strand is for a given gene, the more closely related the species is. Are we all with that so far? Alright, okay, brilliant. Next, this is all to do with DNA, but then we begin to move on to protein analysis. Because as you know, all DNA does. DNA codes for proteins. Therefore, if we look at the proteins in different species, it transpires that we're comparing DNA. Okay? Now, again, what we will do is we will compare the amino acid sequence for a specific protein. Alright? Now, you might think, well, hang on, if it's a protein, it must be the same in all the animals. That's not true. And this is why, it's a very good tool for us to use. Hemoglobin is the classic protein that's analysed. And hemoglobin in different organisms has a slightly different amino acid structure. It will have slightly different amino acids in slightly different positions. Okay? And we can analyse and compare the amino acid chain for a given protein, e.g. hemoglobin. Are you with me there so far? And, again, I'll try and put this onto the board for you as well. If we can have a look. Let's just go to it. Here is another diagram representing different species and their amino acids and we are comparing similarities. I hope you can see on the diagram, some of those species have more similarities in terms of the amino acids in them and the order they are than others. The more similar the amino acid chain, the more closely related the species. Okay? And finally, and this is probably the toughest of the lot, we do immunological comparisons. Now, if you can concentrate on this bit, this is probably the hardest bit. Okay? We're comparing antibodies that are produced by organisms. And to do that, I want to introduce you to a word first. The word is serum. Serum is found in your blood. It is plasma and the proteins that are in plasma. Okay? So serum is plasma and its proteins. Which means serum contains antibodies, which makes it very useful for us to use. And basically, put really simply, okay, we take the species we want to look at. Let's call it species A. Alright? This is the species we want to compare. And we remove species A's serum. So I'm going to get my syringe where I've got the serum inside it, okay? And we take that serum and inject it into species B. Now, we want species B to be very unrelated, because what we want species B to do is to make antibodies to species A's serum. Because it is different, it will recognise all the proteins as foreign and it will make antibodies to them. Alright? So what we're actually making here is the antibodies to A's proteins. Then what we do is we get all the serum from all the organisms we want to look at and add the antibodies to them. So, let's say I'm looking at species C, D and E. And what I want to do is know which of these species is closely related to A. And what I've now got is a syringe that's full of antibodies to A's proteins. Right? Here's the clever bit. If I add those antibodies into each of the species' serum, they will react. There will be an immune response. And there will be agglutination because the antibodies will be binding to the foreign proteins. Do you get that? And the more precipitate or agglutination there is, the more closely related the species are. Now, I'm going to have to do some more detail because you're looking confused. Alright? But if I then put the antibodies to A into these three new samples, the sample or the species that gives us the most precipitate, the most agglutination, must have the closest set of proteins to species A. So if I say this is 90% precipitate, 80% and 70%, I hope you would then say to me, sir, C must be the most closely related organism to species A of the three I'm looking at. Okay? Right, I'll come on to that in a bit.